All right, open up your biology notebooks to your next page and title this DNA Replication Notes. Okay, so today we're going to be learning about how DNA replicates itself, otherwise known as like makes a copy of itself. Okay, and so we saw this a little bit in class, and so now we're going to get into some specifics of how this actually works. Okay, another word for replication is called synthesis. To synthesize something means to make. So DNA replication, we're replicating it, synthesis to make it, so we're making a new DNA, okay? This happens during the S phase of the cell cycle. We haven't talked about the cell cycle yet, but you know how like there are seasons? Winter, spring, summer, fall, winter, spring, summer, fall, and it goes over and over and over again. Well, the cells in your body go through different cycles and they go through different phases and they repeat. And one of those phases is the S phase. S stands for synthesis. Why? Because we are synthesizing DNA. So again, that's a good thing to know because we're going to come back and touch on that later. And during this time, DNA is going to make an identical copy of itself. Okay. So at the end, we have two pieces of DNA that are exactly the same. And just FYI, this takes about 4 hours in a human cell, which is pretty impressive. Okay. And so here's what happens in DNA replication. The way that this works is that we have enzymes are going to help us do this. Enzymes are these proteins that basically do like all the reactions in our body. Okay, so enzymes are going to help with this process too. And you can tell an enzyme by its name because enzymes typically end in the suffix ase. And so we have two enzymes that we're going to be learning about in this process. Okay, there's actually quite a few more, but we're just going to be learning about two of them. We're going to keep it simple. The first enzyme is DNA helicase. See that ASE? You know how DNA is in a double helix? It's in that coiled shape. Okay. Well, what DNA helicase is going to do is it is going to break the hydrogen bonds, see, in between our nucleotides, in between our nitrogen bases. You know how there's 2 and 3 bonds in here? Okay, the helicase is going to come in and break those bonds apart. We can also call it unzipping. If you think of like a piece of DNA like a zipper, like a zipper when it's all zipped up, you can think of this helicase as the part that's actually doing the unzipping. If that makes sense, okay? So again, it's coming in between our two strands, or two halves of DNA, and it is breaking apart those hydrogen bonds, right? 3 between C and G and 2 between A and T. It's breaking those apart so that then we will have 2 separate pieces of DNA, okay? And then what's going to happen is, see, now we've got C over here by itself. And what partners with C? G, always. So there's going to be free-floating nucleotides that are just floating around in your cell. And this yellow enzyme right here, DNA polymerase 3, is going to say, okay, here we go, we got a C. What goes with C? G. And so DNA polymerase 3 is going to grab a G, and it's going to buddy it up with C. And then it's going to go to the next one and say, oh, I got an A. Okay, what goes with a T? All right, where's their T? And it's going to look through the pile and find a T and pluck a T and connect that T to that G. And then it's going to go again. Oh, okay, here we go. We're going to need a C right here. It's going to look through the pile, find a C, attach it on. So now our new piece says GTC, and so on and so on and so on. So DNA polymerase 3, again, this is an enzyme. It ends in ASE, and there's several of these that do different jobs. So we're just going to call it DNA polymerase. But DNA polymerase is going to get these free-floating nucleotides and is going to follow our complementary base pairing rules to add them accordingly. It's also going to do it on this strand down here because remember, these two things were connected. They're going to unzip. And so we're going to have a polymerase here matching up nucleotides and a polymerase here matching up nucleotides. Look how we're still following our base pairing rules. Okay? So helicase unzips. Polymerase is going to make a new strand that is complement to our old strand. Okay? So here we go. Here's our 2 strands. Now they're kind of unzipped. Here are free nucleotides coming in, not pictured to be DNA polymerase, adding them in. Boop, 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 boop. Once we get to the end, notice that we have 2 identical models of DNA, molecules of DNA. Okay, so this is 1 molecule of DNA, and now we have 1 and 2 molecules of DNA. Notice that each molecule is half original and half new. Here the red is shown in new. okay? So you have one strand that was part of our original, and here it's blue, okay? So there's blue, and one strand or one half that's the newly formed, so that's shown in red, okay? So that's a really important thing with DNA synthesis.
To make perfect copies of our DNA, an enzyme first unwinds the two strands of the molecule by separating its base pairs. Another enzyme then attaches free nucleotides to the exposed sites. The existing strand determines the composition of the new strand. According to the base pairing rules, adenine bonds with thymine and guanine with cytosine. In long strands of DNA, replication occurs in many regions simultaneously, forming the sister chromatids required for cell division.
All right, so that was just a nice little animation to show you kind of how that works. You could see that little horseshoey thing that was DNA polymerase coming in and adding our new free nucleotides in, okay? And the last thing we're going to need to know about replication is that there are errors. Like, look right here, okay? If we're supposed to have a C and a G here, but instead there's a T and an A here, that would be an error, okay? Another error would be if C wasn't paired up with G, if it was paired up with some letter other than G. That would be an error too. And thankfully, there are, and remember, enzymes are helpers. There's a proofreading enzyme that comes through and is checking to make sure that everything is matched up as it should be. And if it finds something that is wrong, then it will fix it. And usually those errors are caught, but every once in a while the errors slip through. And those errors, if left unfixed, are called mutations, okay? And we're going to learn more about mutations later, but a mutation can be harmful, it can be beneficial, or it can have absolutely no effect whatsoever. Okay, so that's just a little bit about DNA replication. Again, here we have helicase breaking apart our hydrogen bonds, okay? And then we're gonna have our DNA polymerase adding the complementary base pairs to each strand. So at the end, we have two new strands that are absolutely identical, but each is half original and half new.